ASH 2015 coverage continues. I'm Thomas Baldrick. We're happy to have with us Dr. Michelle Fanale from the MD Anderson Cancer Center. Thank you for taking some time out of your schedule.
Sure, of course.
Let's talk about this study of yours, gene expression profiling for molecular features of response in a phase 1 trial combining Alisertib plus Romidepsin. Why did you choose to do this combination?
So this combination was decided upon because Romidepsin is already approved for patients who have relapsed peripheral T-cell and CTCL, so cutaneous T-cell lymphoma. It's a histone deacetylase inhibitor, and then Alisertib has already been evaluated in a phase 2 trial and now registrational trial, initially for all different types of lymphomas and now uniquely for T-cell lymphoma. So what was known both clinically and then also preclinically is that both Romidepsin and Alisertib work well in terms of a decreasing level of proliferation and inducing apoptosis for T-cell lymphoma cell lines, and then also in the clinical setting for T-cell lymphoma patients in terms of inducing responses. And then there was also some preclinical data that showed that the combination in the laboratory setting was synergistic, and also that it could downstream decrease certain proteins like c-Myc related proteins. And so it was also thought that in addition potentially to being beneficial for patients who had T-cell lymphoma, that maybe it also could work for other types of c-Myc positive lymphomas, such as Burkitt's lymphoma, double hit lymphoma, double protein lymphoma. So there was actually several different reasons to why this trial started.
So how did you take all this knowledge and design a study?
So this trial was designed through the National Cancer Institute through their CTEP drug evaluation program. So within that program, it actually provides principal investigators like myself unique access to drugs. So basically they have a list of drugs where the drugs are basically being provided by the respective pharmaceutical sponsors. You could then design, in this instance, a phase 1, or it also could be a phase 2 trial, where you're using drugs from different pharmaceutical companies in combination. Through CTEP, once CTEP actually approves them the trial, you're then able to get drug supply from those pharmaceutical companies to do the trial, which otherwise might take some additional time. So in other words, if you were going to, say, Pharma Company 1 and then Pharma Company 2 and saying, I want to do a trial that combines both of your agents, there's generally a long time period for contracting and those types of matters, versus CTEP really facilitates this process because they handle all of that through this specialized program.
So another good start. How do you come off in results with this? What happened? What do you have to report?
So right now we've enrolled patients to the first 4 dose levels that are shown in the poster. So we treated patients at dose levels increasing of both Alisertib and Romidepsin, where Alisertib was dosed with the traditional dosing schema. So patients were treated with Alisertib twice per day for 7 days in a row. For the Romidepsin, for the initial 4 dose levels, we gave just 2 doses of Romidepsin. Usually Romidepsin is given 3 times per cycle. We decided to give it 2 times per cycle, with basically the Romidepsin given here and then the Alisertib given in between each of the doses of the Romidepsin. For those initial dose levels where the Alisertib ranged from 20 milligram dose level to 40, and then the Romidepsin ranged from 8 to 10 milligrams per meter square dose level, we saw for the patients who were treated, the best response was actually seen in one patient who had heavily pretreated peripheral T-cell lymphoma, where that patient actually had a prior Romidepsin by itself, and for that Romidepsin that he received by itself, his disease actually increased. But when we gave the combination, his disease actually went into complete remission.
So that person's very happy.
So that person's very happy. We've actually now gone on to redesign the trial a bit. Okay. So there's some preclinical data that came out actually after this trial was already open, showing that to get the best synergistic benefit from giving these 2 agents together, that you should actually do the dosing a little bit differently, so that the Alisertib actually should basically be given pre each dose of Romidepsin and then after. So now within the redesigned part of the trial, we're actually giving now the Alisertib pre each dose of Romidepsin on the day of each dose of Romidepsin and then the day after. And then we also went back to the conventional dosing for Romidepsin, which is actually 3 doses per cycle. And part of that was also influenced by the fact that there were certain patients who actually had disease that was improving initially on the first part of the trial, but then when they had this break in therapy, and they were off treatment for a couple of weeks' time, we saw that their disease was increasing. So we really felt that it would be best to really stick with the traditional dosing schema and have them get that third dose of Romidepsin per cycle.
And how long will this research continue?
So we anticipate that likely this trial will probably take about another year to complete. Based on also this additional preclinical data, we believe that probably the best level of response is going to be seen for patients who have T-cell lymphoma. We know that respectively, each of the agents by themselves induce about a 30% level of response. So we're hoping to see a better level, even than maybe even a 60%, let's say, combined response for the two agents when given with the right dosing format. If that's the case and we see that the best response is in T-cell lymphoma, then we'll actually do an expanded dosing cohort at the maximum tolerated dose level for patients just who have peripheral T-cell lymphoma.
Okay. Congratulations on your effort to this point, and we wish you the best of luck going forward.
Thank you.
Thank you.